Hello everyone, today we're going to be talking about RNA interference. Two very important things to keep in mind is small interfering RNA, siRNA, also sometimes known as short interfering RNA or silencing RNA, and microRNA. Now let's go ahead and cover RNA interference, or also known as post-transcriptional gene silencing. RNA interference is a cellular mechanism that is activated by double-stranded RNA molecules, which ultimately result in the destruction of RNA that contain a similar nucleotide sequence. It is very well known and used as an experimental tool that prevents the expression of selected genes, hence called gene silencing. Okay, that was the basic principle of RNA interference. Now let's backtrack and revisit microRNA and small interfering RNA. We will now focus on microRNAs, and later we will focus on small interfering RNA.
MicroRNA, or miRNA, is derived from a double-stranded RNA, or dsRNA. It forms a pre-mRNA, which is 100 to 120 nucleotides long. It is produced by an RNA polymerase II. Together, DROSA and DGCR8 proteins will form together and produce a pre-miRNA that will form 70 nucleotides long. The pre-miRNA is ready to be guided out of the nucleus and into cytoplasm using the protein exportin 5. The pre-miRNA will be prepared by dicer. The pre-miRNA is being cleaved by dicer, thus forming a 3' overhangs using dicer activity and 2 nucleotides and 5' monophosphate regions. This then will produce a miRNA star duplex. The miRNA duplex is ready to be attached to RNA interface that is loaded in by RISC, that is composed of 2 proteins, slicer and argonaute. During the RISC complex, it is composed of 2 domains, PAS and PB. PAS will bind with the strand and form guide strand and migrate to a target sequence of single-stranded mRNA. The passenger strand or the single strand will then leave. Next, the miRNA will then bind with a target sequence, or target mRNA. Later, the nucleation process will take place, thus forming a double-stranded RNA. This will then leave the mRNA to be cleaved into 2 parts. Afterwards, that means the protein that should be produced from the translating RNA will no longer be produced inside the cell.
siRNA, or small interfering RNA, also known as short interfering RNA or silencing RNA, is a double-stranded RNA molecule with 20 to 25 nucleotides. siRNA degrades mRNA that is no longer needed by the cell, as well as degrades viral RNA to get rid of any viruses in the cell. RNA is cut into 2 separate strands or shorter fragments of 22 nucleotides by using a protein called dicer. According to Christine Enger and Gunther Meister from Journal of Cell Science, the strand takes argonaute, or AGO for short, which are highly specialized small RNA binding proteins. The RNA guides the AGO proteins and combines them with other proteins that combine with the siRNA duplex to form RNA-induced silencing complex, also known as RISC. These RISC are the same ones that carry miRNA. RISC get rid of one of the double strands of the siRNA duplex and use the remaining strand of RNA to destroy foreign RNA molecules, such as viruses, by entering the cell and acting as a defense mechanism, preventing the expression of a certain gene, also known as gene silencing. The authors of Essential Cell Biology state that the RNAi defense response can spread from tissue to tissue, allowing the organism to become resistant to a virus after only a few of its cells have been infected. RNAi resembles certain aspects of the adaptive immune responses of vertebrates. In both cases, an invading pathogen elicits the production of the molecules, either siRNAs or antibodies that are custom-made to inactivate the specific invader and thereby protect the host.
Selena M. Sagan and Peter Sarno knew that RNAi played a role in immune responses against viruses in infected plants and invertebrates. They also knew that in October 2013, some scientists found evidence of a functional antiviral RNAi pathway in mammalian cells. They then had a question: would this RNAi pathway be present in mice? To understand, they knew that virus-derived small RNAs, which are RNAs created by the virus and known as vsRNAs, help the virus grow in numbers. Now, what the immune system does is it creates an RNA-based antiviral RNAi pathway and protein-based antiviral interferons. Now, they decided to use mouse embryonic stem cells, otherwise known as MESCs. And they decided to use two viruses. The first, encephalomyocarditis virus, also known as EMCV. In their first trial with it, they injected the MESCs, and it was noted that most siRNAs remained the same. It was mainly because of the IFN response, hence their function was still not clear. So they decided to use the nodemurovirus, NOV. Let's have 3 mice, and 2 of these mice are 7-day-old suckling mice, and you have 1 adult mouse. Now, for the 7-day-old suckling mice, you have NOV delta B2, which is a nodemurovirus without the B2 protein, and you have NOVM-B2, which is a mutation in the B2 protein. Now, what this protein does, which was in the nodemurovirus wild type, was it would make it possible for the virus to multiply by inhibiting dicer. And it could use ectopic, which means abnormal expression of either B2 or the Ebola virus VSR protein, which can also cancel out dicer's effects in a different way. And so for the first mice, which had the NOV without the B2, and the NOV with a mutant B2, which the mutant B2 wouldn't work, it wouldn't suppress the RNAi pathway, the mouse would actually clear out the virus, which it would do by an RNAi response. However, in the wild type NOV, the mouse couldn't because of the B2 protein, and it would present lethal results. In the adult mouse, for the wild type nodemurovirus, it would use IFNs, which it would actually help the mouse clear the protein. And so this study showed that RNAi pathways in mice is possible. Hence the title of it, RNAi, antiviral after all.